We already talked about the synthesis of novel metal nanomedicines by utilizing reduction processes. Now we're going to be talking about the methods that would be used for modifying the surfaces of those metal nanoparticles to be able to provide targeting ability or maybe increase the stability of these particles in suspension. So to do this, fortunately, we utilize a very easy chemistry that is based on the use of dative or coordinate covalent bonds. These bonds are basically bonds that are formed where, so let me back up. In a typical covalent bond, what happens is that two atoms are going to share electrons, and the electrons that are shared come from each of the atoms. In a coordinate bond, what happens is that both electrons actually come from the same atom. And so in this particular case, we're going to be using the electrons, the unshared pair of electrons from the sulfur atom of a thiol group to be able to form a dative bond with the metal atom on the gold particle or silver particle. The thiol group is this sulfhydryl group, which is what's typically utilized. So basically, in this particular case, we're going to be making our particle by the reduction of chloroauric acid in the presence of, in this particular case, tetraoctyl ammonium bromide and sodium borohydrate. But we're also going to include something like a polyethylene glycol chain that has a terminal sulfhydryl or terminal thiol group in order to provide the ability to functionalize the surface. Even after we have made that particle, or in whatever form you have formed the particle, let's say that we have these particles that are stabilized with citrate molecules, either of those particles could then be further modified by adding other sulfhydryl containing polymers or sulfhydryl containing chains to modify the surface. So in this particular case, we might be modifying it to include a fluorophore and then to include a targeting agent or something like that. And we can do it in sequential steps after the fact. And basically the reason for this is that this thiol to metal bond is not as strong as a true covalent bond, and that enables us to be able to displace prior molecules that are here. For example, you can see from this particular representation to the next, we actually displaced the original surface modification by adding a new one. So because the bond is actually not as strong as a covalent bond, we can substitute or displace whatever was already there by utilizing other molecules that also contain the thiol groups in their terminal end. All right. So this is, again, very easily, basically what you would do would be to put a suspension of your particles in the presence of the molecule that you are trying to attach to the particle. As long as you have those sulfhydryl groups or thiol groups, then some of that would actually end up on the surface of the particle. Now, this is great in that it enables you a lot of flexibility to be able to functionalize the particle after the fact, but it also brings the question of stability. If this is if this can be displaced very easily, then what's going to prevent it from being displaced maybe before the particle has reached the location where it needs to reach? So before the targeting agents are able to attach to their target, or before the whole particle it comes out from the blood vessels into the site where it is needed, right? So in order to overcome this, what some researchers have done has been to utilize what's known as a polydentate thiol molecule, where we have not just one binding site from a sulfur to the metal, but maybe two or more, and that's what we have on the next slide. So here we have some images of multidentate thiol molecules, and I'm going to go ahead and write that down here. So multidentate, meaning that we have more than one binding site. And of course, examples of that begin here. This is number 1, 2, 3, etc. And what you can see is that now we have two sulfur groups that enable the formation of these dative bonds to the metal surface. And that's the same thing here and here. Of course, in this particular case, we have the two sulfurs that are next to one another. At the very bottom here, we have other examples, number 5, 6, and 7, that include kind of linker groups that separate the thiol groups from one another. As you can see here, we have one thiol here, another thiol here, which enables the formation of that dative bond with the surface. On the left side, I do have a monodentate modification. For example, if you want to provide a primary amine functionality to the surface of your gold particles, then you would utilize a molecule like this one that it's NH2, CH2, CH2, SH, and that enables the modification as such with a single sulfur. And similarly, you could generate almost a self-assembled monolayer by utilizing chains, like longer chains of hydrocarbons that would kind of stack up with one another, forming that kind of a very packed, dense structure on the surface. And of course, by utilizing different chain lengths, you could potentially have a presentation of targeting agents or something that you would attach on these carboxylic acids, which are outside of the length of the original ones here. All right, other type of nanomaterials that are utilized are magnetic nanomaterials. Among those, superparamagnetic iron oxide nanoparticles, also known as spions, are among the most widely used. These are about less than 15 nanometers of size, and they have unique paramagnetic properties. While the larger iron oxide particles that you might be familiar with are ferromagnetic. So paramagnetic means that these materials become magnetized in the presence of a magnetic field, but their magnetism disappears when the field is removed. So basically, you are able to manipulate these particles in the presence of a magnet, but when the magnet is removed, then or when the field is removed, they are no longer presenting these kind of properties. So that's important for them. The reason why these particles work as paramagnetic is because they have a single magnetic domain, and this is as opposed to the larger particles that would have several magnetic moments or several magnetic domains that can be aligned and that produce a permanent magnetization. These superparamagnetic iron oxide nanoparticles are made from iron 2,3 oxide, which is typically written as iron 3O4, and the applications of these materials are going to be in a number of different areas. One of them is in bioseparations. So for example, imagine that you are trying to capture a biomolecule from a complex physiological fluid like blood. And so what we want to do is to be able to use an antibody that can detect or bind to that molecule, but we want to be able to remove it from the solution so that we can get rid of all of the interfering cells and molecules that might be also present in that sample. So because of that, we can use gold nanoparticles, or not gold, I'm sorry, we can use the spions and modify them with antibodies, either directly or through polyethylene glycol chains, and enable this to interact with the physiological fluid. The antibodies would then bind to the target. Let's say that our target was these red spheres, and we can then apply a magnet and capture the particles that now have the target of interest on them. And we can remove the supernatant, which would contain all of the other competing or interfering molecules that we don't want. So that's in bioseparations, and this is a great application in the preparation of biosensors, because the first step in a biosensing is to be able to separate the molecule of interest. We can also utilize spions for targeted drug delivery. In this case, we would actually use the magnetic properties to be able to force or encourage the particles to go to the location where we want them to go to. So for example, let's say that we're trying to target these particles to cancer cells. We could maybe inject the particles near the location where they are needed, and then we could try to kind of encourage them to transport to the right location by using a magnet that would guide them in the right direction. We can also utilize them for biomolecule or cell manipulation in different applications. We can utilize them as contrast agents for MRI. In the case of MRI, spions actually provide negative contrast, And that's what you can see here. So on the top, we have spion labeled cells, and the arrow is pointing at a dark spot. That dark spot indicates the localization of these particles. So if you have functionalized your particle perhaps with antibodies that are able to bind to a tumor, for example, then the particles would provide a dark shade on the location of that tumor. And then we can also utilize them for magnetic hyperthermia. In magnetic hyperthermia, basically the particles are exposed to an alternating magnetic field, and when they attempt to align their magnetic moments in that field, that produces a lot of friction that then leads to the formation of heat. And for hyperthermia, basically what we're looking for is the temperature going from 37 degrees, which is normal physiological fluids, to now being something in the range of 43 or higher 43 or higher temperature, which can basically cause damage to the cells. These magnetic nanoparticles can be utilized by, can be synthesized by precipitation or microemulsion processes, or they can also be synthesized by high temperature precursor decomposition processes. The next type of particles are quantum dots. These are very small, usually less than 10 nanometer nanocrystals that are made from semiconductor materials. And the main property that we utilize in biomedicine from these quantum dots is their fluorescent capabilities, the ability to provide light, which can obviously be utilized for bioimaging or biosensing. Typically, these quantum dots are made from cadmium selenide with a zinc sulfide shell. They can be prepared by a number of methods, one of which is high temperature precursor decomposition, where you basically would begin with compounds that contain cadmium or selenium or zinc or sulfur, and you would expose them to high temperatures in the order of 400 degrees Celsius or so, and enable the formation of these quantum dots. Let's see, one thing that is really important about these quantum dots is that the band gap size is size dependent, which means that their optical properties can be tuned kind of similarly to gold nanostructures. And that's what's shown here on the right hand side. Basically, the size of the particle is what controls the band gap, and the band gap is what controls the optical properties or the place where these particles are going to fluoresce. So, for example, here we see that we have particles of different size, 2 nanometer versus 2.5 nanometers versus 3, and so on and so forth. And you can see that as we increase the size of the particle, of the quantum dot, the wavelength at which that particle fluoresces is redshifted. And so it provides us great tunability. And one other really key thing about these quantum dots, as opposed to maybe organic fluorescent molecules, is that they have much higher brightness, orders of magnitude, like 10 times to 100 times more brightness in terms of the amount of fluorescence that they can provide compared to an organic molecule. And they're much more stable, meaning that they're not going to be photo-bleaching, meaning that they can withstand excitation by a light, a laser, without their optical properties diminishing. And this is very different from organic dyes, which typically would decompose or would photo bleach upon exposure to light for long periods of time. These gold nanodots, these quantum dots, I keep saying gold throughout the whole presentation, I apologize. These quantum dots also have very narrow and symmetric fluorescence spectra, which is nice because it enables you to do multicolor fluorescence labeling, for example. So in a single cell, I could use different quantum dots to be able to label different parts of the cell. Let's say that I'm interested in protein A, protein B, protein C. I could then functionalize these quantum dots onto antibodies that are specific for each of the proteins, and I could see the localization of each of those proteins independently, just because of the fact that the fluorescence peak is so narrow, and I'm able to separate the optical signal from each of these by utilizing the appropriate optical filters. And the last thing that is really cool about this is that they can be excited with just one set of light. So I can expose my cells to, let's say, ultraviolet light, and I can collect the fluorescence from all of these simultaneously, as shown here. In this particular case on the top, the vials that contain the quantum dots are being exposed to ultraviolet light, and you can see the fluorescence from all of them at the same time. That's different from any organic molecule where you would have to excite each of them according to the peak where their absorbance is. And then, so you would need independent excitation and independent detection for each of the organic molecules. That's not the case with quantum dots. So from the standpoint of the optical properties, these are really great for bioimaging. The problem is the toxicity. Many of them are known to be toxic, and while there are some methods to minimize that by maybe encapsulating them within silica shells and other methods, their actual use in biomedicine is very restricted. It's definitely only restricted to in vitro assays or biosensors that are external, not for in vivo in human patients. Some of the applications that have been utilized is in optical barcoding, because again, you're able to utilize multiple labels in a single sample, and for molecular imaging. All right, and that takes us to silica nanoparticles. So silica nanoparticles are typically prepared through this method called the Stover method, which is a hydrolysis process. In this hydrolysis process, we begin typically with a silane precursor, such as TIOS in this case, which is tetraethoxysilane. You can see tetra meaning 4, ethoxy, that's this OCH2CH3, and then silane because the core, it has the silicon structure. And this molecule is hydrolyzed, basically leading to the formation of alcohols. In this particular case, it's showing one of the 4 ethoxysilane components coming off, leading to this alcohol. And then once this happens on several molecules, the alcohols can come together in a condensation process to form the SiO-Si backbone of the polymer. This can, of course, occur not on just one side, but on all sides, possibly, leading to the formation of a polymer system. Now, these kind of nanoparticles can be easily fluorescently labeled, as shown here on the right-hand side, utilizing organic dyes, for example, by inclusion of the molecule called APTES, or aminopropyl triethoxysilane, which is basically a silane precursor that incorporates a primary amine. That primary amine can then be labeled with a fluorescent molecule or a dye through chemistry that in this particular case is showing the N-hydroxysuccinamide ester chemistry, where these two come together forming an amide bond in between. And I should say that the amide bond is not being shown here. Actually, it is here. Here's the amide bond between the two. And then this can be, so you could actually incorporate APTES at the beginning during the synthesis process of the particle itself, such that at the end you have the amine group. Sorry. Or you could pre-make the silica nanoparticle and then post-functionalize it by adding APTES. And I should say that all of these hydrolysis and condensation processes are catalyzed by either base or acid, and depending on how it's done, you might end up with particles of different geometries or different sizes. In this particular case, I am actually showing some images from prior work that I did with silica particles, and we did obtain highly monodispersed particles that were spherical or that were actually flat, kind of like pancake shape, depending on what kind of methods we use for their preparation. I apologize. So one key thing about this silica nanoparticle preparation method is that it really produces nearly monodispersed particles compared to the preparation of other types of polymeric nanoparticles, for example, which you guys actually got experience on in some of your experimental projects. Also, the nanoparticle size and morphology can be controlled by the reaction conditions, including the concentration of the precursors or the inclusion of a catalyst. You can also obtain porous silica nanoparticles by introducing a surfactant such as acetyl trimethylammonium bromide, which are going to form micelles that get incorporated within the silica network or silica structure. And after the fact, you could use calcination in order to remove those organic molecules, basically by burning them off and leaving just the silica porous network that initially was surrounding this surfactant. That's known as the modified Stover method. What else do I want to say? Silica nanoparticles are known to be biocompatible. They are fairly easy to modify, as I mentioned here with the APTES example. They can be porous, and they have applications in drug delivery, gene therapy, bioimaging, and the work that I was actually doing at this point in my work was to utilize them as agents for biosensors, where we were trying to make them to be fluorescent in the near infrared range. So here we're actually seeing pictures of the particle suspensions being irradiated with a near infrared light source, and the fluorescence was actually not visible because it was in the near infrared, so it was not visible to our eyes, but I was able to capture those images by utilizing a camera that had its lens removed to enable the capture of photons in the near infrared range.